it's okay because I wasn't going to show so many data slides tonight, but I just wanted to first of all start by again recognising the work of Peter Rees-Evans, who was a friend to many people here in the room, including myself, and clearly we've all been shocked by his passing, but we'll do everything we can to remember him and to make sure that his legacy lives on. Mm-hmm. So that's a really important thing for me and for others in the room, no doubt. So what my talk is going to be about, if it appears, is about, it's in the programme I think about immunotherapy and virus therapy. I'm going to spend most of the time actually speaking about viruses, mainly because I love them, and secondly, because of course our lives have been turned upside down by one of these blighters over the last couple of years. So I thought it would be useful just to give you an idea that actually there are some uses for these things, despite the fact they can cause lots of disruption. But before we start with that, I'm just going to talk a little bit about immunotherapy and what is immunotherapy. So one of the most commonly asked questions, certainly in the first decade of my career as a consultant, was, what can I do to help my immune system to fight this cancer? To which I used to make the usual sort of bland remarks by, take a bit of exercise, eat well, and fingers crossed something will happen. And of course, in the meantime, while I was making those rather bland remarks, people were doing really important work in laboratories around the world. And we were actually working out how the immune system fails to engage with cancer, because the cancer evolves as part of its life cycle, in part of its development. It learns how to silence the immune system. Thank you. It learns how to silence the immune system, and it has specific mechanisms. I've got no slides about this, Sarah. I'm just going to go with it. Thank you. So it learns how to actually disengage the immune system from being able to attack the cancer. And so having understood how the immune system does this, or how the immune system is put out of commission by the cancer, we were able to start designing drugs that could actually recommission the immune system to be able to re-engage with the disease. And I'm pleased to say that amongst the other tumours, including the poster child of immunotherapy, which is melanoma, amongst other diseases, solid cancers, including head and neck cancers, lung cancers, kidney cancers, a range of other tumour types, including really difficult to treat diseases like mesothelioma and anaplastic thyroid cancer, we now understand that we can use a class of drug. And so you hear this word immunotherapy, by which most people mean one of two classes of drugs, either an anti-PD-1, anti-PD-L1 is the other variety of that, or an anti-CTLA-4 drug. Now these drugs have all got ridiculous names, and the abbreviations even for them are rather ridiculous, but essentially these are what are called immune checkpoints. And an immune checkpoint is something that a tumour can learn to use to keep the immune system either quiescent or worse, actually to kill the immune system. But by using drugs that re-engage those pathways, we can actually allow patients to have better outcomes. So in the context of head and neck cancer, we at the Marsden, and with support from Oracle Cancer Trust, we've been able to be engaged in a number of the leading clinical trials that have led to changes. So going back to 2016, we published a paper with colleagues. It was a large randomized phase 3 study that for the first time led to the approval of a drug called nivolumab to treat patients with relapsed head and neck cancer who had already had one go at chemotherapy in relapse, and then they had immunotherapy, and we showed significant improvements in survival for those patients. Having achieved that, we were able to say, why don't we bring that immune therapy into the first line treatment of patients with relapsed cancer and test a drug called Pembrolizumab, either on its own or in combination with chemotherapy against the gold standard chemotherapy. And in a phase 3 clinical trial published in 2020, we were able to demonstrate that both single agent pembrolizumab and the pembro-chemo combo, because we love a rhyming abbreviation, the pembro-chemo combo was better than standard of care chemotherapy. Not only that, it was significantly less toxic. Patients lived longer and they had fewer side effects. And remarkably, in a paper that will be published next month, we show that for for patients who have a certain level of biomarker, a protein in their tumour, those patients, and that constitutes about 43% of the population, for a patient population that has this high level of biomarker, the survival at 4 years is as high as 30%. Now, when I started treating patients with relapsed head and neck cancer, when Chris and I took over the practice, there was one man who used to come to the clinic, and all the registrars would be introduced to this gentleman because he was the only person we'd ever met who'd lived more than 2 years with metastatic head and neck cancer. We now see routinely patients returning who have been in remission for 3 and 4 years. Not enough of them, of course, but enough cause to give us some optimism that we're really going to make a difference. So there's a whole panoply of new immunotherapy drugs being developed, and I'm not going to talk to you about them tonight, but be happy to talk to you about them in the Q&A session afterwards. And I'll mention them very, very briefly, just with some pictures at the end, because I'm going to risk showing some slides now. So I'm going to talk about viruses. And so the first position to take, of course, and this is what's often something we have to address with our ethics committee and our GM safety, our genetically modified organism safety committee, is that viruses are scary. And I used to say viruses are scary even before the pandemic, and now, of course, they're really scary. But even before the pandemic, you can see from these dreadful photographs, some of them rather pretty electron micrographs of viruses, which are geometrically very lovely, but in terms of what they do to human beings, they're absolutely awful. So if you look at the one up in the top left-hand corner as you look, which is variola major, that's smallpox, which thankfully is now extinct in the world until someone deliberately releases it from a laboratory, God forbid. But variola major is estimated to have killed more people than all the wars that have ever taken place in the world. And this is just one of many viruses that can cause problems. You can see that we have to face a number of new emerging viruses. So for instance, the Zika virus was unknown pretty much until less than a decade ago and is now causing major problems. And I don't really need to remind you about that virus, coronavirus. We knew about coronaviruses, and of course there was an episode of SARS about a decade before this pandemic. And now, of course, this particular version of coronavirus has really put us all on watch that we need to be careful about viruses. But that's not the only thing that viruses do. They cause 25% of all cancers. And of course, unfortunately, there are people in this room who know that only too well. There are 2 viruses here on this slide. The Epstein-Barr virus, it's associated, amongst other things, with nasopharyngeal carcinoma, and the human papilloma virus, particular flavors of this, particularly HPV-16, is associated with head and neck cancers. But they're, again, one of the largest killers of patients with cancer, the hepatitis viruses, which thankfully there's traction against these now, hepatitis C being potentially a curable disease. But there are a range of viruses that can cause cancers. But again, I don't want to dwell on that. I want to dwell on the fact that we can use viruses to treat cancer. And so in the last 3 or 4 minutes, maybe a little longer, I'm just going to talk you through some of the ways that that works. And I would really encourage you to go and speak with Vicky Ralston, who will be standing beside a couple of posters in the room next door, who has really done some wonderful work on this, funded by Oracle Cancer Trust and funded by the Mark Donegan Foundation, to which I would like to pay tribute. So we have worked over the last number of years with a whole range of different viruses, and there's some weird and wonderful things here which hopefully you've never come across. The one that you might have heard about most obviously is HSV at the bottom, which is the Herpes Simplex virus, which is the cold sore virus, which causes irritating eruptions of cold sores just when you're about to go on holiday or a big day to a job interview, all those sorts of things. But we have worked both in the laboratory and in the clinic with all of these except for SFV, which is a very pretty virus called Semliki forest virus, and SVV, which is the Seneca Valley virus. So people go out into the wilds, they collect dead animals, or they get ditch water, or they get bits and pieces of the ground, and they take them back to labs and they isolate viruses, and then people look at what those viruses do. And a number of these viruses do a really remarkable thing. And And they do something that looks like this cartoon. And this is what we call oncolytic virus therapy. So normally, when we get infected by a virus, it will go into the cell, it will try to grow, because that's what a virus needs to do. A virus is not alive until it's inside a cell, formally. Although we talk about live viruses, they're not live until they're inside a cell. But the cell, our cells, have evolved an immune system that shuts off those viruses, stops them replicating. And it does this through various mechanisms, including things like the interferon response. And so the virus will be shut down in a normal cell, most viruses. And for an extra measure of safety, the cell that's infected will do the decent thing and it will commit suicide so that there is not an ongoing infection and the immune system can come and clear up the debris. And that process is called apoptosis. But these remarkable viruses, some of which I showed as pictures on the preceding slide, there are some viruses that can do really interesting things when they find themselves in a cancer environment. Now, you'll notice in the cancer environment that little red stalk that I drew at the top, which was the antiviral response, isn't there. Because one of the things that cancers do to become cancers is they often shut off their immune response or their interferon response so that they can silence immune responses against themselves. But it means that this virus now finds itself in a fertile soil where it can grow, whereas in the normal tissues and the normal cells it cannot grow. And the orthodox view is it makes enough of itself that it pops the cell, and those viruses will go back and infect a neighboring cell. And so it goes on, and the idea is that you give cancer the flu, and the cancer would be killed by a wave of virus infection that would sweep through it and eradicate the cancer. This is what we thought 20 years ago. It's not true. We get relatively low levels of replication in truth in patients, but what you really trigger with these things is you trigger an immune response. And again, if you can bear another cartoon, and I'm not sure how well, in fact the body, there is a body here actually, but it's disappeared. It now looks like disembodied lungs and lymph nodes, but there is a form there, I promise you. Imagine this patient's got a tumour about here. I can almost see the legs there. Anyway, the patient's got a tumour. You inject the tumour with some virus. The virus infects the tumour. It grows in the tumour cells. It pops those tumour cells. It bursts, releasing viral proteins, proteins that you encode within the virus. So you can genetically modify the virus to make it express things that can rev up the immune system if you put the right things in there. But it also makes the tumour release a bunch of proteins that it would rather hide inside itself, and these are called antigens. So you make this tumour now very visible to the immune system. You recruit some cells, which are called dendritic cells, which will hoover up all the debris, and they migrate to the lymph nodes nearby, where they go and they show these protein fragments that they've hoovered up, and they tout them around to cells in the lymph node. It's a bit like someone trying to sell something in a marketplace, but what they're showing is there a T cell here in this lymph node that can see this particular fragment of protein that I found in the tumour, and the tumour was trying to hide from you, and if there's a T cell that sees it, those T cells will get activated, they will replicate, there will grow millions and millions of these T cells, which will then be released into the body, into the bloodstream, and they will go and hunt down tumours all around the body. And this is a cartoon, of course, you've seen lots of cartoons tonight, but we've demonstrated that this can actually occur within melanoma. We have enough data that led to an FDA approval and an approval in the European Medicines Agency, and even NICE decided to fund it, so it must be good. So we've got an approval for a virus therapy for cancer. And again, as I say, go and talk to Vicky at her poster. She knows all about this virus, but more importantly, with the support of Oracle Cancer Trust, we're also able to develop new viruses. And I'm not going to talk you through the diagrams that indicate how we've manipulated the genome of these three new viruses called RP1, RP2, and RP3, because we're not very imaginative in the way we name things. But essentially what you see here is a range of viruses which are already in the clinic. RP1 in randomized phase 2 trial, RP2 in an expansion study in a phase 2 study, and RP3, we're just completing the first part of the phase 1 clinical trial at the Marsden Hospital and elsewhere, including colleagues in Oxford. But what all of these viruses have done, and you can see there are lots of different coloured bits, and the genome's been chopped about and has been rearranged. We've made viruses that will make, within the tumour where they infect, they will make these immune checkpoint molecules. So RP2, for instance, makes a drug called ipilimumab that costs about £70,000 for a course of treatment. And this virus actually makes it inside the body from the raw ingredients that are present in the body. Now, I suspect that the drug company will probably factor in that £70,000 into the price of the agent, but nonetheless, these things are very clever agents. And these things are now all being tested in patients with a range of tumours, and I'm absolutely delighted to be able to report that RP3 is being deliberately targeted now for patients with squamous cell carcinomas of the head and neck. Head and neck is seen as a big target. There will be a trial both in the relapsed metastatic setting in combination with chemotherapy, but really importantly in combination with radiation therapy at the time patients are first diagnosed with cancer. And that's going to be specifically for head and neck cancer patients. I said I'd say just a few words right at the very end about new immunotherapies. And so, as you might have worked out, these don't look very much like new immunotherapies. They're just ways of me reminding myself what I wanted to tell you about. So this partly eaten burger is to remind me to say that there's a bunch of treatments being tested now that try to block something called the don't eat me signal. So cancer cells, as a means of protecting themselves against the immune system, are actually able to display a protein called CD47, which actually shuts down the cells that would come in and chew it up. So we can now block that signal with anti-CD47 antibodies. That's in randomized phase 2 clinical trials. We lead that trial at the Royal Marsden Hospital. The thing in the second picture is a bee sting. And so we are working on a number of things called stimulators of interferon genes, the sting agonists, and we're running 2 clinical trials. Those things are injected into tumours. They mimic viruses. They're not viruses, but they mimic viruses, and they cause interferon release and they tickle up the immune system. The third picture, which look like very nice mustard coloured, I don't know what those things, they look like Watsits really, don't they? They're not Watsits. Those are Yersinia enterocollitica bacteria. And we're going to do a first, John, you were going to say that.
Yeah,
I could hear you. Those bacteria in a first-in-human study, first time these will ever have been given to a human being, we hope to do that within the next month at the Marsden Hospital. These things have been engineered. They have little proteins on their surface which are like hollow hypodermic needles, and they can inject into immune cells proteins. Now, when you get a disease with Yersinia enterocollitica from eating a bad piece, usually of pork, this bacterium gives you very bad illness because the bacterium is able to kill cells of the immune system by injecting them with poisons called YOPs. We've taken those genes out of the bacteria, and instead we've replaced them with genes that will stimulate the immune system directly. And so the idea is that you have an engine for driving the immune system. And then the final picture at the end, which is a bunch of skyscrapers, is to remind me to tell you about a whole range of different immune checkpoint inhibitors. And this is the Skyscraper 9 study. There aren't 9 skyscrapers, I couldn't get quite the right picture. But the Skyscraper 9 study is using a new immune checkpoint blocking drug called an anti-TIGIT antibody. So there's sunshine shining through there, through the top of those skyscrapers. And so I think it's really important that we should be optimistic about where we're going. There's a whole bunch of really exciting new treatments. Richard is right. We shouldn't be spending huge amounts of money just at the end of life. And ideally, my vision of developing these things is not to use them when patients have already had 5 different lines of treatment, but to bring them right up front. Treat the patient at the very first time of diagnosis, cure them the first time they present with disease, because then you never have to treat relapsed disease. And that's what we are aiming to do, and obviously with better quality of life. So I'm going to close there.